Lega e per la stanza invece questo. Se c'è qualcosa io guardo da casa in cose. Perfetto, poi comunque anche se non sono vicinissima mi sentite, no? Facciamo il Dolby Surround?
No.
Ah, guarda, scusa,
Dico sempre che lasciare che devo ripasso il
sì.
lo gestisci come vuoi, compri se ce l'hai radio e
Sì, adesso infatti alla fine volevo parlare con
ti organizzi tipo anche loro, anche come si fanno le donne.
certo. Distrazione, infatti.
Possiamo andare, ragazzi? Sì, ci siamo. Sta registrando? Sì.
Sì, tutto partito.
Quindi oggi chiudiamo l'intervento di Marina Grazia Quero, la settimana della didattica. Dopodiché, spetta a voi, diciamo, il compito di fare l'esercizio che vi abbiamo insegnato ciascuno di noi. Fatto prima, entro il 12 giugno, ce lo riconsegnate singolarmente, e dopo noi restituiamo il nostro giudizio. E ammesso o non ammesso, tutti anche ammessi, e se c'è nel giudizio magari possono anche metterci in maniera che si rendano conto di avere più scontri che abbiamo fatto sentire. Marina Grazia ci presenterà il lavoro che lei fa sulle attività costiere, prevalentemente,
sì.
Immagino, attività costiere lunari, e quindi tornerà sul discorso del problema che in qualche modo un pochino ha accennato a donata prima. Quindi mi raccomando, studiamo bene anche questa lezione, già su questo tema ci sono ancora ragazzi che ci stanno lavorando, quindi devo avere la massima attenzione. Faccio i lavori di fuori dentro, proprio il laboratorio, la propria attività, e ci rivediamo prossimamente con gli appuntamenti che abbiamo avanti. Il 20 marzo abbiamo il primo turno dei 38esimi che discuteranno la testa finale della difesa. Anche una commissione esterna e straniera a noi, quindi abbiamo che si confronteranno con, oltre ad aver avuto i revisori che hanno rivisto la testa e che hanno dato il loro giudizio, da una commissione esterna. Chi vuole il 20 marzo può assistere qui, guardi quello che adesso per essere in conto dei studianti. Sei sempre preparato. Buon lavoro, grazie.
Grazie Mauro, grazie mille. Grazie a voi. Okay, thank you. The last effort for this week. How many of you are microbiologists? Uno, due. Okay, so I will try to keep it simple as much as I can. I'll try to be not too much technical, just in case you can interrupt me and ask for specify or talk again about one of the aspects I'm going to present. Okay, we are talking about microbial pollutants. Microbiome, as Mauro was mentioning, is you know better than me this week, no? Marco also explained some days ago. It's the full community of microbes living in an environment, okay? Today we are talking about a specific part of the microbial community, which are the microbial pollutants, bacterial microbial pollutants, to be more precise. The lesson will be divided in 3 parts. So we will make a short break between one and the other, also because I'm not used to talk for so much time in one day. So we'll give you an introduction on microbial pollutants at sea, an overview of case studies on which we have been working or we are currently working. The third part will be a short overview of methods to be used to study, which are currently used historically or as an alternative to historical methods to analyze the presence of microbial pollutants in the sea. And the very last part, it's just a few spots on initiatives like research program fundings, some website which can be of interest for you, also in light of what we are going to do after the PhD, okay? So imagine that even if this is not properly a topic in which you are working on, maybe in the future will be something you will work with to integrate your research, okay? So if you're working on pollution, on chemical pollution, on plastics, on fish, it's very likely that this topic can be also associated with your research, okay? We will talk a lot about fecal indicator bacteria today, okay? I think that, are you all Italians? Or there is someone from outside? You. Maybe in the last year, you've heard, last year or last 2 years, you've heard about news talking about beach closures, especially in the Adriatic side of the sea, no? The bathing water often experience, in the last year more often actually, levels of contamination by fecal indicator bacteria which are above the limits. This is happening more and more frequently, and we will somehow talk on the reasons why. And this is not only true in the Mediterranean Sea, but also at the international level. So this is becoming something that we are very interested in the last years also, because it has many consequences of on tourism, on health. We will see, because they are directly linked to bathers' health. It happened to us at CNR to be contacted many times in the last years to make interviews about this. So people want to know why this happened, what does it mean, why we cannot go to swim in our beach, in our coasts. So it's something that really touches us very closely. So when we are talking about microbial pollutants, we are talking about microbes, and this means bacteria, viruses, or fungi, which are of human or animal origin and that are released in the sea. Today we are going to talk about bacteria primarily. They are mainly attached to particulate matter, so they like not to be free-living most of the times. And that's why there are some kind of sources which are particularly related to the release of microbial pollutants at sea. They are the responsible of bad water quality, especially in bathing waters. And the problem is that they can accumulate in filter feeding shellfish. So there is a risk posed by food or animals living in the sea. And the water, the contaminated water itself, can also transmit potentially diseases to bathers, okay? Microbial pollutants can reach the coastal environments through very, very different ways. This is a very simplified scheme, let's say, reporting the main sources. Most of them also, the ones included in the case studies I will present later, are represented by rivers discharging in the sea, which collect basically contaminants or contamination through its way to the sea. So from agricultural waste, from sewage or not properly treated water entering the river and then the sea. Aquaculture can be a source or a sink of microbial pollutants. Plastic waste can be the so-called hitchhikers of microbial pollutants. So bacteria like to create biofilms on surfaces, on plastic surfaces, including potential pathogens, and then it can be transported in the sea. Ballast waters are also a way through which microbial pollutants can be also transported along high distances, okay? Of course, hospitals and discharge from hospitals which are not correctly treated, can also represent a way to introduce microbial pollutants at sea. And of course, cities, tourism can be also very highly contributing to the presence of microbial pollutants. What I've been working mostly is microbial pollution through rivers and through discharges at sea. You can see from the pictures here that even a small, relatively small river can introduce to the coastal sea. Imagine during the last year, we are experiencing a lot of very heavy rainfalls events, no? This is causing a lot of water from the rivers, including suspended matter, to reach into the coastal sea. And most of the times, these phenomena are also linked with the release of higher abundances of microbial pollutants. I'm talking about microbial pollutants, but within this term, we are talking about different types of microbes which are historically or more recently detected to define the quality of water, okay, including seawater. Most of the times we are talking about microbial pollutants, we are talking about the traditional fecal indicator bacteria, the FIB, we mention them as FIB, which are, we will see, represented by Escherichia coli and Enterococci. We have also, for several reasons that we will also see in the next slides, the research has been working on searching for alternatives to the traditional ones because they have pros and cons, okay? They have limits, they are powerful in some ways, but they don't give all the information that we usually need when we are going to microbial pollution. And so we have also these so-called alternative fecal bacteria and sewage-related fecal bacteria, which have been defined by several works of Dr. Newton's group, and they are mainly targeted through DNA-based applications, so with molecular techniques. We have also a very large container of the so-called MST, which is the microbial tools tracking. It's basically a set, an increasing set actually, of molecular target of genes of bacteria or viruses which are specifically associated with the animal host. So we will have targets indicating the presence of fecal contamination by cows, by dogs, by seabirds, by humans, etc. So we can also understand where this contamination is coming from. And finally, we are also, especially in our works, we are also looking at other potentially emerging pathogens. There are studies all over the world indicating that there is an increasing incidence of some bacteria, for example, Acinetobacter, Pseudomonas, and some taxa, which are becoming very alarming for the unexpected presence and resilience in the marine environment. And finally, the last part will be of this overview will be dedicated to antimicrobial resistant bacteria, so bacteria of natural origin or released in the environment possessing genes for resistant to antibiotics, okay, which are transmittable to other microbes. Okay, the first category is the fecal indicator bacteria. So fecal indicator bacteria, which are E. coli and Enterococci, are a very small subset of the so-called fecal coliforms. Of course, they are coming from, most of the times, from feces. We will see that some of them can adapt to the environment, but most of the times, I mean, the theory behind using them is that they come from feces. And that's also why E. coli is the most studied bacteria in the history of microbiology. No? We have like millions of papers and research regarding E. coli. It's very easy to isolate him. We can find them in many types of environments, but the primary habitats is the warm-blooded animal gut. Okay, so that's why it's used together with enterococci as fecal indicator bacteria. It's very easy to, they are very easy to cultivate and enumerate, and that's why they are still used despite their limits as globally as indicator bacteria. They come from warm-blooded animals, so they are adapted to temperatures of about 37 degrees. Okay, so keep in mind this, because they will go from certain condition from the warm-blooded animals gut to the condition in the environment, which are very different, especially those in the marine environment. Okay. E. coli and enterococci can actually be found also in secondary habitats, so soil, sediment, and water, and this is also a reason why they can be enumerated in the environment. When we are indicated them as fecal indicator bacteria, because the problem of finding them in the in seawater, in the marine environment, is not their presence alone. I mean, E. coli and enterococci come from, most likely, from feces, but they are not necessarily pathogens. Okay, so they can be completely innocuous. Most of the E. coli in our gut is commensal, so it's used to live there, is not harmful, it's not pathogenic. Some just of them can be pathogenic, but the idea is that if we found them, and they are very easy to enumerate, okay, even technically, if we found them in seawater, it's very likely that there is a fecal input, okay, a fecal contamination from other sources. That's why they are called indicators. They can indicate the presence of other potentially more pathogenic, more dangerous, more harmful bacteria, okay. Do you know the regional agencies for environment? No, the ARPA. The ARPA in Italy, the other agency in EU, most of the environmental agencies in the world use these two indicators, or at least one of them, to define the quality of bathing waters. And the quality is established considering some threshold. Okay, so if you go and take seawater sample and you find in marine waters more than 200 CFUs, which is colony forming units, in 100 milliliter of seawater, the quality of water is not sufficient. Okay, it's bad quality, and that's the case in which a beach place is closed. Okay, when we are over the threshold, that is basically closed until the concentration of these bacteria go again to the below the limit. E. coli and enterococci have different concentration allowed for reasons related to statistical studies. They have different threshold also in fresh water compared to seawater. And this is also something that the research has been working on for decades. I mean, it's since the 1920s that fecal coliforms, including E. coli and enterococci, are used for these purposes. I would like to show you the Portale app. Do you know this website? Have you ever worked with this? This can be very useful also in your everyday life. Portale Acque, and also the regional website for Marche is a free website from the Ministero of the Health Ministry. You can find here all the measurement of fecal bacteria. There are a lot of parameters, but you can find the history of the measurement of bacteria of FIB in all the coasts of Italy. So the monitoring season usually goes from May to September, so waiting season, summer basically, and you can go there and find all the monitoring sampling and results and see if your favorite swimming place is clean or not. For example, in Ancona, you know that there is often the closure at Passetto, no, and some of the place close to Passetto. You can check why on the Portale Acque. I would like to show you briefly the website here. This is the national one. Okay, you can look for your place. For example, here, you will see that you have these segments, which are basically the sampling point. They are always the same, okay? The region, the APA has a monitoring program for the bathing season, basically, and you can click on maybe on one of them, and you will see the results, okay? You see the last available data, so 2026 is still not started. You can go here, you can see the trends and the dates of sampling. You can go to the previous years. You can see, these are the threshold, okay? You can see when the sampling was done. You can see the numbers of cells of FIB found in that place. You can download data, actually. And so you have a lot of very nice information to check, okay? You can see that here, in this case, in Fano last year, you never were above the limits, okay? In case ARPA finds high levels, so non-acceptable levels, the sampling is increased in frequency until you reach again the good quality, okay, the sufficient quality at least.
There is a sensor that runs the analysis, or they sample every day?
They go in these dates. They go there. They take.
Every month.
Yeah, it depends on the monitoring programs. In some places, there are also more frequent, like only every 2 weeks. Because the regulation wants you to go there, take seawater samples and play them on growth medium and use a particular technique. Yes, you have to go there. And we still don't have a very reliable sensor for, yes, that can detect specifically Enterococci and E. coli. There are some, but they cannot be used for applied EU regulation, so they are not accepted. You can go for research purposes, but not for official monitoring plans. And you should have a lot of sensor along the coast, okay? They're expensive. You should go there with the sensor at least, no? So the technique that we use today to do this, it's still very cheap compared to all the other techniques. That's why we will see in the third part of the presentation.
You go with the boat and sample the water?
It depends on the site, because you have to follow some indication bathymetry. So you have to take water when you have at least, I think, 1 meter of depth. I don't remember exactly. This is our first job, yes. But it's very, very costly. Sometimes they go entering in the water without the boat.
Because I can see those spots, they are part of it.
In this case, yes. And I think this is related with the bathymetry. It depends on the place, yes. For example, here you see, yeah, it's very close to the coast. This is the regional website. It contains the same data that you saw in the national one. It has a different graphic version, and you have also other information about not only the FIB, but also some environmental parameters that can be also used to do research on this data. Okay, I will show later a study that we did using more than 10 years of data produced by ARPA on the Marche region to understand whether we have trends of increase of FIB along the Marche coast, because this is a research topic which is being explored all over the world. Okay. 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 So there are limits of using traditional FIB, which are, I think I'm working on this topic since 2013. And there was already a discussion whether these FIB were still reliable, okay, in the science community, in the scientific communities. We are still using them at the UN level. Some Australia and US are using molecular tools to detect them, so they are applying alternative methods and alternative targets to detect microbial pollution at sea, but we in Italy and in Europe still use FIB to understand the quality of our bathing waters. Okay, the limits are mainly, bacteria are very good in adapting to new conditions. Not all of them, but some of them can. So some Escherichia coli can adapt to the environment. Some enterococci can, but most of the time we have studies regarding E. coli, can survive, can adapt, can grow in sediment and in seawater, can grow on algae. And this means that if they are naturalized or adapted to the marine environment, what is the meaning of using them as indicator of human or animal contamination? It doesn't make sense because they are already in the environment. So we are measuring something that is already in the environment. Okay. They are not able to tell us what's the source of the contamination. So E. coli and enterococci can come from cows, from humans, can come from birds, from other domestic animals. We are not able to understand the source. We know that there is pollution, there is contamination, but we don't know why or who is causing this. And most of the times, to find a solution to contamination level, we need to know what's the source. Okay, it's very important to understand where the contamination is coming from. And this is one of the points which is most of the times used to push towards other methods and to push towards other targets to detect microbial pollution in the ocean, in the coastal sea, is that the current methods used is very time consuming. So when we will see the cultivation method that we use takes 24 to 48 hours before having knowledge about the contamination level. Meanwhile, if people is in the sea, in a contaminated water, this is risky, because if the levels are too high, there is risk of illness, there is risk of transmitting microbes to, I don't know, mussel aquaculture nearby. So 24 to 48 hours is not a short time. It's enough to have risks, okay? So we are trying to, scientific community is trying to push towards faster methods. That's why other microbial pollutants have been identified. One is the alternative and sewage fecal related bacteria, which have been basically identified analyzing tons of molecular data. Okay, in the last 20 years, we are producing a lot of DNA sequence data of microbes, of microbial pollutant, or microbial communities, especially in the sea, and not only. So we are gaining a lot of information about microbial diversity, and we have enough statistical power to understand whether some taxa are associated or not with some types of environment, okay? That's the theory behind what the group of Dr. Newton has done. They have basically identified statistically some groups, even at higher taxonomic level. So we don't have the species, we have maybe orders or family of bacteria, which are which can be used most reliably as species associated with respect to E. coli and Enterococci, and some of them, such as Acinetobacter, Arcobacter, and Trichococcus, which are statistically associated with sewage infrastructure. In sewage, you have like biofilm created on the surface, okay, of pipes, of tubes, and most of the times you will find these microbial taxa there. So they are very associated. They found there for several reasons, including particulate matter accumulating in this structure, the favorable condition to thrive, okay? And that's why what we are doing also in our group is to look to entire microbial community data and to search for these targets as novel and alternative indicator bacteria of fecal contamination, okay? And this is one of the most, in my opinion, the most interesting application, microbial source tracking. This term includes a lot of things inside. In case of marine environment, is defined as a method, molecular method to target fecal microorganisms. Okay, so fecal microorganisms which are specifically associated to a host. That's why we have pieces of DNA, pieces of genes which are associated with a specific host. So far, we have. I think this is just. I don't know if you can read it, but we have genes that we can target to check for chicken, for duck, for swine, for cattle, for human. So this is a very powerful tool to understand this. You can quantify the microbial pollution in the site that you are studying, and you can understand who is providing contamination. A final description is, final part is on antibiotic-resistant bacteria. In this big box of microbial pollutants, antimicrobial resistance is today one of the most important challenges, okay? There is a global fight. There are scientists working together all over the world to fight antimicrobial resistance, because this is becoming a real problem. It is already, and a real problem at the global level. Okay, so bacteria which can resist to one or more antibiotics. In the sea, what's happening is that we release not necessarily antimicrobial resistant bacteria, but genes of resistance. Okay, so piece of DNA which were included in a bacterium which was resistant. Once they are in the environment, they can be acquired by some of the bacteria living in the environment, which can be those released by human activities, which can be those already living in the marine environment. Some bacteria can acquire resistance genes, and this can be translated in a risk for both marine biota and humans, okay? So antimicrobial resistance gene in general is a very hot topic also in the marine environment. I will talk later about aquaculture and antimicrobial resistance. because it's something that we have been working on. Many of you knows Naomi, which also had, she's finishing a PhD on this topic. So she worked for 3 years to understand whether aquaculture was a simple source of antimicrobial resistance. Okay, even in this case, antimicrobial resistance, and I'm talking about both bacteria and just genes, just DNA, can enter the marine environment through very different ways. We have evidence that antimicrobial resistance is present all over the world, in every part of the ocean. And we have to keep in mind that antimicrobial resistance is a natural phenomenon. Okay, so Mike, I don't know if you heard about a very recent news about a research group in Romania. I think they found a multi-resistance bacterium in a cave. Okay, and so it was not a contamination by humans. It was there. It was living there. Okay. And this can happen. Antibiotic resistance is a natural phenomenon. So it's just a strategy of bacteria to survive to stress outside. So but they are able to exchange. And the more antibiotics we use, the more we stimulate the acquisition of antimicrobial resistance. That's the problem. That's why we should be careful in using antibiotics in everyday life, including in aquaculture, when antibiotics can be given to fish to fight for infections. Okay. So I was mentioning at the beginning of the presentation that microbes, frequent indicator bacteria, let's say for example, E. coli and Enterococci, comes from our gut. In our gut, the condition is a very high temperature, so 37 degrees on average. They are very concentrated. They don't have oxygen, and most of the time they don't have salt, let's say, no? It's not a saline environment. So when they go to the marine environment, especially, they found in the best case scenario, 20 degrees. In winter, maybe it's even lower. In several places, it's even lower. There is more oxygen, and there is a lot of life, including competitors, including predators, let's say, so other organisms which can feed on them. And so they found a very, very unfavorable condition of the environment. Okay. The marine environment in particular, it's different from, let's say, inland waters, so from rivers, from lakes, which are rivers, lakes, because we have a lot of hydrodynamics, we have currents, we have continuous exchange and movement of water, we have UVs, which most of the time is a killer for bacteria. We have salt, we have different temperature, and so there are many reasons why microbial pollutants should not be there. But there are some of them, as I was mentioning, which can adapt. Okay. This is a summary of something that we will see later in the case studies. This picture is showing macroalgae in the Lagoon of Venice. In some part of the year, they produce amazing amount of biomass. Okay, so it's so incredible. It's so the biomass is so huge that sometimes boats, public transport is blocked because they cannot navigate into this biomass. Okay, it's a lot, and most of them are species which have been transported with ballast water, so they are basically invasive. No, we saw it was studied in some lakes in the US, and we saw this even in the Lagoon of Venice, that macroalgae, for example, are a very good place for fecal bacteria and even for naturalized one to thrive. Okay, to to live, they have food, they have protection, they have surface, a lot of surface. So the environment can offer opportunities of adaptation for some of these microbes. So most of them will be transported, will die in a matter of a few days, if not hours, but some of them can still adapt to the environment. And in my opinion, this is the most interesting part of all the story. So if you want, we can stop for 10-15 minutes. I don't know. Le 3:10? Ok. 3:20, 3:20, 3:25, riprendiamo?
Yes.
Avete domande? Interrompetemi quando volete. Sì. questo e questo, giusto?
Sì.
Lo posso mettere giù anche, no?
Sì, sì.
Ok. Okay, let's start again with some case studies. This section of the presentation is very biased towards my work, so you are going to listen about what I've been doing and I'm doing now with the research group also at CNR in Ancona. Maybe. Okay, some works that I'm going to present focused on microbial contaminants in the Lagoon of Venice. The title of this slide is a natural lab to study microbial pollution because it's true. I mean, the Lagoon of Venice, I've been working there for several years, so Giorgio knows very well the area. It's a beautiful place, a unique place in the world, but it has also many impacts insisting on the environment, okay, in the lagoon. The building, the city has been built in a very peculiar way. I don't know if you know that it's basically there is no land below the buildings, the historical buildings. It's mainly created on trees, which have been put in the mud and that sustain the buildings of the city. This means that the architectural part is also very much linked with some of the reasons causing microbial pollution in the city, okay? Because you have to imagine that the sewage system and the purification systems of house waters, it's a bit different than that of normal cities, let's say, okay? This is one aspect. The other very well-known aspect is the huge amount of tourists visiting the city every year. We are talking about hundreds of thousands, if not millions of people, okay, which are causing several types of impacts. They also contribute to, let's say, a stress on the sewage system of the city, okay?
If you fall into the channel, you have free blood analysis.
I never tried, and so I'm not sure they are going to do. No, I think that the word free it's not applicable in Venice. No, I think you have to pay. Yes, maybe you are fluorescent after. You're like a superhero, but I'm not sure. Sorry. Yeah, you have to pay to enter. So it's. I mean, it sounds very weird to me. Yes. So tourists, you have also a lot of traffic, of boat traffic, okay? The lagoon is on average very shallow, maybe, I think the average is 1 meter, George, I don't know if you know at this. You have some shallower parts, you have some deeper parts, but on average you have 1 meter depth. And this means that the continuous movement of the boats, of public transport, of big ships, both commercial and touristic cruises, are causing a lot of resuspension of sediments and the continuous mixing of the water, okay? So it's very easy that all the lagoon becomes mixed with everything. It's causing pollution, both chemical, biological, microbiological, etc., okay? Plus, this is one of the most parts of the city, the tides, the so-called acqua alta. The lagoon is separated from the Adriatic Sea by a very thin part of land, which is open in 3 parts. There are the so-called bocche di porto, mouths, which connect the Adriatic Sea to the lagoon. And this allows for a continuous exchange of water between the Adriatic Sea and inside of the lagoon, okay? this mechanism, which has a 6-hour cycle, so you have a tidal cycle of about 6 hours, which allows this movement of water from the sea to inside and the opposite, contributes often to let's say the renewal of water within the lagoon. But the city is so peculiar, I mean the building, the architecture is so peculiar that some part of the city are like going down into the mud, okay? And this means that sometimes the tides, when the level of the tides is particularly high, and this is usually happening from in winter, let's say from October to February, more or less, you have exceptional tides, which are also influenced by wind, by weather condition in particular. You have some part of the city is completely flooded, okay? And I mean, this creates also, there are some studies by American people. Some years ago, there was a study in particular, which made a collection of viral and microbial pathogens in the water of Venice. Okay, so these people here is literally doing swim in a like in a cultivation broth or something like that. No, I would never do something like this. I never did, I swear. But this is, I mean, this is just to say that apart from the craziness of these pictures, the tides have also some influence on the concentration, on the abundance of these microbes in the waters of the lagoon. Okay, this is a section, is a scheme that I found on a very, let's say, popular website explaining, I'm not able to explain you in detail, but this is how the sewage system in the houses of Venice is generally done. There is a discharge, and the water in some part of the cities, in some building, was, I mean, the system was renewed, was updated, let's say. But not all the city has a modern system. This is, I mean, this is impossible because the historical buildings cannot be modified as we want. So there are some constraints to the historical buildings to even for renew the sewage system. Okay. So guess what? We found PIP, so microbial pollutants in the water of Venice. We have generally found in different places, in different period of the year, rather high levels of, especially of E. coli. We found up to 10 to the 4th cells. If you remember, the limits for E. coli was 500. Okay, so we have rather high peaks in the abundance of these cells. Okay, we didn't found a relation with physical chemical parameters. So our hypothesis is that it's not very related to what's happening around. It's more related to the continuous input that you have from the buildings. Okay. This study was done in different places. You have to imagine, if you know the city, you have wider canals, you have narrower canals. Some of them are more deep, some of them are very shallow, some of them go toward an open basin, for example, the one of San Marco. So you have very different situations, okay? And this can also somehow influence the levels of FIP that you can find there. But a very interesting part of this study was that in any case, so in all the sites that we were studying, we found a very nice relation between peaks in FIPs and lowest point of tides. So there was an inverse relationship between tides and abundances of E. coli. What we imagine is that when the water, when the tide is entering, there is, let's say, clean water coming, and there is a sort of dilution of the bacterial abundance. included also the microbial pollutants. And when the tide is low, I mean, the level in the canal is lower, and this means that this discharge, the volume in which the discharge is has been input is lower, so the microbes are more concentrated. And so there is, I mean, tides is also an influence of this. If you really want to go for a bath in the waters of lagoon, go when the tide is high, okay, at least, because maybe is less risky. I take advantage of this slide. You see that we have the graphs for water and for sediment. Everything I've been talking about, speaking about feed contamination, so regulation of feed done by monitoring programs, etc. So by the official regulation, the official protocols, is done on seawater. Okay, so the matrix which is used to define the level of pollution in the marine environment is water. The sediment is not included in this analysis, but sediments usually host one order of magnitude more bacteria than the water. Okay, so naturally it has a very protective environment. It has a lot of organic matter. It has, it can be protective towards UV, towards biota, towards other organisms feeding on bacteria. So it's very good for bacteria, and this means that also microbial pollutants, including FIB, are generally higher in sediments, although they are not targeted in official regulations. Okay, we found that this was true even here in the Lagoon of Venice. So there was generally higher abundances of FIB in the sediments than in the water, but sediments are more stable. So this link between peaks in tide, so this inverse relationship between tide and abundance was not observed in the sediment. In the water, it was very clear. Okay, high tide, low abundance; low tide, high abundance. This was not true for sediment, and maybe because at least in the places that we use for sampling, it was like not very suspended, and it was like a more protective environment for E. coli. This is my, one of my favorite aspects, which is the adaptation of Escherichia strain, not necessarily E. coli, to the environment. So this issue was raised by several research by the Dr. Work. He defined the cryptic Escherichia strains. Cryptic means that in the environment, in freshwater environment, actually, he found Escherichia, so members of the same genus of E. coli, which were phylogenetically distinct from the ones coming from the mammals, okay, from warm-blooded animals, and that were shown to be environmentally adapted. So they were living there in the environment. They were not released by humans or by animal feces. They were living there in the environment. This is why we still are questioning the use of FIB as indicators. Okay, what's the problem? The question is whether the environmentally adapted, so the naturalized Escherichia strains are more able to acquire virulence or antibiotic resistant genes. So if they can represent a risk, so once they become adapted, whether they represent a risk for human and for marine biota as well. Okay, we have not completely clarified this. Actually, we have several clades of cryptic Escherichia. Some work on the virulence and antibiotic resistance acquisition capacity has been done on one of the cryptic clades, I think the E. But most of this topic is to be explored, completely explored. But it's particularly interesting to understand both the reliability of FIB as indicator and the risk posed by naturalized fecal bacteria. This is the same picture that I showed you before. Again, in the Lagoon of Venice, we found evidence of the presence of these cryptic clades. Okay, we searched. We were looking for them both in water, in the sediments actually, and on the surface of these macroalgae. And what we found is that they go selectively on algae, okay? So they like that environment, it's protective, it's nutrient rich because it's full of polysaccharides, whatever they need. We also tested them in laboratory condition to confirm this hypothesis. And basically, we have already evidence of naturalized and environmentally adapted strains, okay? We didn't check for the presence of virulence or antibiotic resistance. It's a rather old study. We are currently working on this because we are working on a data set of E. coli genomes from all over the Adriatic coast to see the distribution of antibiotic resistant genes according to their let's say the phenotype, including the presence of cryptic lineages. So we will see. This is a study, it's more recent. We did this template of water and sediment along this transect. So Venice is this fish-shaped part in the middle of the lagoon. Here you have one of the biggest mouths towards the Adriatic Sea. So we basically sampled water and sediment from outside, close to the city, towards the inner part of the mainland. Here there was like an historical industrial site. There is partially working. Marghera, yes. And this sampling was done because there was this colleague from the US, Stephen Techman. He was going around the world to sample microbiomes in the most important ports in the world. Venice port, it's not one of the biggest, but it was particularly interesting because it's very, very, I mean, it's a globally recognized important sites, no? Let's say, for tourism, for history. And so he also decided to come to Venice to do the same kind of research. And what we did was to use microbiome data, so sequencing data of the microbial communities collected in both seawater and sediment in all these sites, and look for those targets that I was mentioning at the beginning. So sequence recognized as E. coli or Enterococcus, so the traditional one, the alternative fecal bacteria, the sewage associated, and a list of other potential pathogens that usually are considered as such all over the world, okay? We found evidence even from this data set that microbial pollutants in the wider sense, so including all the different categories that I've been describing, they like particle attached fraction. So they, in this data set, there was like 2 different filtrations, so on filters with different pore sizes. The one describing the microbial pollutants, the free-living microbial pollutants, and the other one was to describe the particle attached one, okay? So we kept the particulate matter, basically. And we didn't find microbial pollutants on the free-living part. So the focus for them, the preferred place for them was particulate matter, okay? This means that when you have discharges from sewage, which is very rich in particulate matter, if we find a way to disrupt this particulate matter, maybe it's already a way to reduce the contamination reaching the sea. Okay. We already knew this, but we found evidence even in this data set. I will go directly on the last part. What we found, of course, was what we expected. That is that most, based on the data that we have from microbial community, so from sequencing data, we had some indication of the sources, okay? And the main source, of course, was human. So what we see in the reality was also measured by, by this study. Man is causing most of the microbial pollutants in the Lagoon of Venice until the inner part. So from the very inner industrial port through the city until also some miles outside the lagoon. Okay, we still found sequences of microbial pollution. We found differences. You see these big balloons. These indicate different types of microbes, of microbial pollutants, and in some cases we have it was a bit tricky to find an explanation. And for example, in this case, we found a lot of sequences of sewage-associated bacteria, but as far as we knew, there was no sewage system there, as far as we know. So maybe there is some leaking or some, I don't know, something not official discharging there. We don't know. So we couldn't explain this. In the other cases, the data that we found were quite reasonable according to the possible inputs of microbial pollution. Also, just to tell you how clean is the water and the sediments of the city, we conducted a very small experiment with some students from an American university in the sediment. We collected sediment of in this part of the city, and we looked for antibiotic-resistant genes. And we also found antibiotic-resistant genes everywhere. Okay, this was not quantitative because we analyzed metagenomes, so it's molecular technique that it's not quantitative. We have an idea. We can have relative quantifications, but we still have to be careful in describing the data, but we found evidence of antimicrobial resistance in all the sites that we had. This is, expectable, no? There is continuous discharge into the canals. There is continuous resuspension. There is, spread of microbes, of material, of particulate matter all the time. Okay, so this was expectable. The nice thing, nice to say, let's say, is that we also found evidence for heavy metal resistance genes. For some molecular genomic reasons, antibiotic resistant genes are often associated. If you find antibiotic resistant genes in a genome, you will likely find also heavy metals resistance genes. And both antibiotic resistance and heavy metal resistance showed the presence of resistances to widely used antibiotic and metals which have been historically used and impacting the lagoon. So we had also from the microbial data evidence of the impacts insisting on this very fragile site. Okay, ciao to Venice. And let's go to the Marche region. The summer is approaching, so let's see if the seaside in the Marche region is safe or not. Okay, of course we have different levels of impact here in the Marche region with respect to the Lagoon of Venice. Okay, we are in a very different place. It's a touristic place. So we have a lot of people here, but I mean, Venice alone makes a lot more people than market usually. But we have industries, we have some historical contamination site, we have agriculture, we have livestock, we have some, and we have a lot of rivers, which discharge also, they collect potentially pollution of different types and discharge it into the sea. So one of the first works that we have done here with most of the study I'm showing also involved Marco Basili, so he's also responsible for this. One of the first study that we did was to understand whether different rivers with different let's say putative or not putative or real levels of contamination were also giving microbial contamination at the sea. So the sampling strategy was the one you see here. We basically went in front of the mouth of the rivers of the Esino, the Kent and the Tronto, considering the Tronto, of course, the most polluted ones of the three. And we collected seawater and sediments from close to the coast offshore, okay, in order to see whether we could find signals of contamination even far from the river mouth. And the answer was yes, basically. So if you remember the list of possible sources of contamination I presented maybe in the second or third slide, it also included rivers. And it's true. So even in case of relatively small rivers, we found them that they are contributors to the microbial contamination in coastal environments. We found, even if at different levels, the presence of fecal contamination, so different fecal target, and also other potential pathogens. If you see here, maybe you can see the labels, but in general, the more polluted in wider sense was the river, the more the higher microbial pollution we found. So the Tronto was the most polluted, of course, even from the microbial point of view, and the other two were at different levels, were much lower impacted at the microbial level, but depending on the site, they contain more potentially pathogenic genera versus fecal contamination. The other thing was that we found some positive correlation with some nutrients, which are also, of course, released by rivers. But the strongest correlation was with some polycyclic aromatic hydrocarbons. So the question is, since they are positively correlated, does this mean that the sources of chemical contamination is also the source of microbial pollution? I don't have the answer. The very nice thing to do would be to use microbial source tracking. So the molecular techniques that targets host-specific genes of fecal pollution along the rivers, and see whether in the different places you are going to sample along the river, you can find a different level of contamination and the source, so that you know, for example, that in proximity of livestock, I don't know, a farm, you find a lot of contamination, so high level of contamination by the host-specific target, and maybe that's the reason of the contamination, even when you go to the coastal environment. Very quickly, this study was done also using DNA, microbial DNA. So we relied on DNA sequences we got from the microbial communities. There are and maybe Marco showed you something, I'm not sure. There are a lot of bioinformatic tools to exploit as much as possible single molecular data. For example, we work on 16S data. 16S is a gene which is present in all the bacteria, which is used as an identity card, let's say. So the sequence inside, once you get it, you will match it with a database and you will give a name, let's say, to the unknown bacterium, because it's very similar to unknown sequence, okay? Exploiting this, bioinformatics can give a lot of information. In this case, we also try to understand whether this data could also give more information on the sources. 16S alone cannot give you the source, but there are some statistical / bioinformatic tools that can predict or infer the source. And in this case, yes, we were quite convinced that the main source was human. So it was the sewage, the infrastructure collecting sewage from humans that was mainly causing microbial pollution. Okay, this is a study that we performed not so much time ago with the ARPA Marche, so with the regional agency. They provided us with more than 5,000 measurements of FIB collected along more than 10 years, yeah, 11, on this portion of the region. So from Senigallia, basically, to the south of Ancona. Data were collected, so were available between May and September for these 11 years, because in the rest of the year they don't do monitoring, it's not required. And we started this study because in some parts of the US, especially in Florida, if I remember correctly, there are studies indicating that there is an increase of fecal contamination over the years. And the question is, does this have something to do with climate change, with increasing temperature, with increasing, let's say, strength of rainfall events, of other weather events? And we try to answer this question even in this portion of the post. So what we found is that FIB contamination changed over space and time. I'm not going into detail of the differences between the different portion of the post that we analyzed, but I can say that Senigallia is usually very, very clean, Ancona a bit less, Ancona, some part of Ancona. But this increase was not an increase in abundances. So we are not seeing this trend over time, okay? What we see is that along these 11 years, there is a reduction of the so-called structural zero. So it's less likely that you go to the sea, you sample, and you will measure zero, okay? You will find something, maybe a very low abundance of fecal indicator bacteria, maybe below the limits. So the overall quality of the water is okay, but it's less and less likely that you will find zero. And this maybe is, indicating a trend of something. We don't know. So it's a statistical increase, let's say. If you're interested in statistics, there is the methods part here, which was developed by an Irving colleague, Stefano Guicciardi, which is an expert, and you can find specification on this. It's very difficult to deal with this kind of data set because they are full of zeros. Okay, so it's particularly challenging. The question was, do we have other data that we can try to relate to these changes in abundances over time? And for this aim, we used river discharge data for a couple of rivers, because it's very difficult to get this data, I must say, rainfall data, and CSOs. CSOs are combined sewer overflows. These are, to make it very, very simple, there are these structures collecting water from sewage system, but when the rainfall event is particularly heavy, so you have a lot of water insisting on this system, they are not able to contain all this water, and the municipality allows for the opening of a door, and the excess water goes into the sea. Okay. And basically, in this portion of region, river discharge was not explaining peaks in FIBs, okay, because we had these graphs showing peaks. Rainfall did not explain fecal pollution peaks, but these overflow events did. So what we found is that every peak in FIB concentration corresponded to the opening of the sewer. When you have the opening of the sewer, you could or could not have the peak. But if you had a peak, it was for sure connected with the CSO event. Okay, so maybe at least here the main cause of this excessive loads of FIB in the coastal environment is represented by these openings. Okay. I must say that also retrieving data on the openings by municipalities was quite challenging, because there is not the system for which they are activated is not automatic. Okay, there is a very old style system of phone call. I will open. You don't have a register. I mean, it's quite difficult to find this, but this can be a point to be improved in the next years, I guess. No, so we published this with the ARPA, so they know that this was the. I mean, we discussed this together, and we agreed that this can be a very important point to discuss in order to have a better quality of water, even when we have a lot of rainfall. No. Okay, aquaculture. Aquaculture. So I work at the IRBIM. We work a lot with fish farms, with experimental aquaculture, and we look, of course, at the microbial side of aquaculture. Okay, both in beneficial terms and in potential contamination by dangerous microbes or by antibiotic resistance. We did this work examining what we know so far in the Mediterranean about antibiotic resistance and aquaculture, and the picture in the Mediterranean and in the world, it's very fragmented and it's very different. So aquaculture has huge biomass, huge numbers of fish in a cage. Okay, so if there is an infection in one fish, it's very likely that it will spread very fast because they are in contact. I mean, they are living in a very close environment. They are close to each other, and so the infection can spread very quickly. We have been working with a commercial site some years ago with Marco. We were going there like every month to make samplings, and they experienced a huge mass mortality event of fish because of a bacterial infection, Photobacterium. And it's as far as they told us, and it was moving from the south of Tyrrhenian Sea. They were in Tuscany. And basically, I mean, the sea transport everything, also bacteria, and this bacterium was able to move. Maybe also infected fish escaped. But they had, I mean, we could not go there anymore because they had huge loss of biomass, and it was they told us it was very, very challenging to go there and remove the huge amount of fish, dead fish. Okay, we are talking about very high number of fish. 2021 forse? 2021. Yes, because we started the cycle in 2020.
But the study is still under review or?
No,
Ah.
this has been accepted last week.
In aquaculture?
Yes. You can, yes. Yes, there is the published version there. But I mean, this is just to say that in some cases it's necessary in aquaculture to use antibiotics. So in the northern shore of Mediterranean, so in the European countries, consumer sensitivity to antibiotic-free food is very, very high. Okay, so most of the times, and most of the plants we have been working with, also on the Croatian side, they are very careful in maintaining the antibiotic-free status because it's a commercial added value. Okay, so antibiotics should be used only for specific reason. This is very different in the southern shore. We don't have the same regulation. It's very different in Asia. So in Asia, intensive farms are using a lot of antibiotics, even to prevent infection. Okay, so there is a completely different way of dealing with the use of antibiotics. So in general, regulations, farming practices, species, farmed, etc., are all topics which can really change our view of antimicrobial resistance in Mediterranean Sea. The question is, the Mediterranean is a semi-enclosed basin, so what you do on the southern shore can have consequences in all the basin. So antimicrobial resistance, as I was saying, it's transmittable. Microbes, water moves, microbes can be transported by water mass, by animals, whatever. So it's not confined in the place you are using it. Okay, if you give a selective pressure by antibiotics in a place in the sea, it can be impacting also a very distant place. I will just show some results of this project. This was a JPI funded project, and I will talk a bit more about the JPI initiatives later. The project Arena was done to understand whether two sites of aquaculture were considerable as sink or source, okay, of antibiotic resistance. And very, very quickly, we found residues of antibiotics, residues and genes of antibiotic-resistant genes contamination in all matrices that we target there, and I mean fish muscles, fish skin, sediment, seawater, clothes, I mean in correspondence to the fish cages, water entering the RAS system, so the recirculating aquaculture system. So we found evidence of contamination by antibiotic-resistant genes and antibiotic everywhere, even if we were working on antibiotic-free sites. Okay, and this, I mean, the resistome, so the full collection of genes of resistance were by anthropogenic inputs. Okay, so basically, both in water and in sediments, although in different ways, we found antibiotic resistance. Okay, the nice thing, and this reflects what I was mentioning before, is that sediments, even if it contained less diverse resistance genes, it contained more sequences, and this somehow suggests that sediment, even for resistant genes, is somehow a reservoir. Okay, so in general, sediments are very interesting to be studied in terms of contamination, microbial contamination, also in terms of antimicrobial resistance. We had indication of resistances to antibiotics, which are not used to aquaculture. So basically what we found in the sites that we have worked on is that aquaculture was a sink. Okay, so there was not use of antibiotics. There was mainly contamination coming from the environment around. Two things: the marine site. So this picture refers to a very small aquaculture plant. It was on the coast. Okay, you can go. I mean, like the coast is here, and the first cage was there. It was very coastal. Okay, and it was very close to a small harbor and urban discharge. So it's very likely that the source of contamination was represented by, I mean, the context around. And this poses question to also to where is the most safe area where to initiate an aquaculture plant. Very different. Very different was the context of the RAS system. RAS is they were like land cages taking water from a brackish lagoon, very close. And the RAS system can have benefits, can have advantages, but it's also very sensitive to the quality of water entering. Okay, so the quality of water was already not very good. There were a lot of birds flying around the system, which also contributed to microbial pollution, and so we found there a lot of more of antibiotic resistance. Break. We can have the last break and then the last part. Sono le 16:14, tra un quarto d'ora? Va bene?
Sono pubblicato sul...
Questo qui, l'ultimo no, stiamo scrivendo
quello
adesso, sì. Questo
no, quello pubblicato su Aquacalcio.
Sì, quello è disponibile. Sì.
Ah, non l'ho trovato.
Adesso te lo...
Ho cercato su Netflix, ma non l'ho trovato.
Guarda, ti dico... Siamo tutti? Possiamo aspettare ancora qualche secondo? Chi è che dà il comando? Allora, aspetta. Non sarò io forse si è preso un attimo di pausa, non sarò io a rovinare la tradizione. Mi sa che è molto paziente, Greta. Ti accolla anche tu, mi sa, tutte e due. Chi è che- ma infatti l'avevo sentito, sì, l'avevo sentito. Tutto è registrato, eh? È tutto registrato. Deve essere quando? Post. No, sono stata io, Greta, non me la sentivo. No, dai, Okay, last part, methodologies. Please stop me whenever you want. Sorry, for the microbiologists, this will be very basic. So, now it's just a quick overview just to show you what we can do, well, which are the official methodologies. I said already something before, which are the alternatives and what are the potentialities of, especially of molecular methods, some molecular methods that the scientific community is developing in the last years. Okay. So we will. I will show you two main categories of cultivation-based methods. Of methods, one is cultivation-based, and the other one is DNA-based. I will show just the most important techniques. So I mean, the one dealing with cells and the other one with dealing with DNA molecules. Okay. So the culture-based methods, maybe you have heard about it during university, I think, if you're all biologists. This is a very classical approach to enumerate, isolate, characterize sometimes microbes. So basically we use the so-called Petri dishes, which are these containers on which there is a growth medium, okay, which is solidified by agar, by a gel, okay. The medium contains what is necessary for the bacterium, which is targeted by that medium to grow, okay. We have hundreds of media for several purposes. They can be more or less specific to isolate certain bacteria, which can be E. coli, enterococci, like in the case of these two pictures. I wanted to bring you a couple of them, but we didn't have available in the lab, so next time. We have those selecting by the presence of antibiotic specific resistant bacteria or not. We have those with a particular color indicating by the change of the color some bacteria performing a particular process, etc. In this case, I'm showing the two plates that we really use for enumerate FIB, so Enterococci and E. coli and Enterococci. They are colored like this. And basically what we do is to take, this is what ARPAT does, basically. They take seawater, they filter seawater onto a membrane disc, okay? The membrane disc, the membrane filter has 4 sides of 0.22 microns, which is enough to stop all the microbes on the surface. So everything that is bigger than that size, including microbe, and everything else, so the rest of water and smaller particles, smaller compounds flow through, okay? So you concentrate millions and millions of bacteria on a very small surface, and you put this on another plate and let it grow, incubate at and temperature, an incubation temperature very similar to the one of our body, so 37, 40 or 42 degrees, it depends on the medium, for 24, 48 hours, okay? And what you see is basically that your clean filter, which contains cells, becomes full if it's contaminated by FIB, becomes full of these spots. Each of these spots is called colony. Okay, this basically, in theory, created by one single cell, which it's constituted by copies, millions of copies of the same bacterial cell. Okay, you need this because unless you let them grow, you will not be able to count them. They are microscopic. Okay, so this is a trick to count what was the number, the original number of cells in the sample. Okay, the blue one, this medium, which is the one mostly used at the European level, is not selective for E. coli. So you have E. coli and other very close bacteria to E. coli. But the E. coli are recognized because they are blue colonies, okay? The other one are light blue, reddish, pinkish, so not doing the same substrate conversion that does E. coli. And then you count them and you know in 100 milliliters how many of them there was. And you can see whether you are below or above the limit. This is why the table I showed you at the beginning sets the limit of fecal indicator bacteria to colony forming units per 100 milliliter. This is one colony forming unit, okay? So you cannot talk about cells, you call them technically colony forming units, okay? This is the same for Enterococci. You basically count the red, dark red colonies, and you will have the number of Enterococci. It has a lot of advantages to count FIB in this way because you have numbers, you have absolute numbers. You can quantify the number of cells there. It's very cheap. So compared to all the other systems, is very, very cheap. You can isolate colonies for further analysis, for example, to see if they have genes for virulence, pathogenicity, antibiotic resistance, whatever. And but they also have disadvantages. So one was the time needed, so 24 to 48 hours, as I was mentioning before. And what is actually growing here is only cultivable microorganisms. So if in the sea, in the seawater you are taking, there are stressed cells, so stress which are trying to struggle with the unfavorable conditions, they don't have enough nutrients or whatever, they will be not able to grow here. So it's very likely that you are underestimating the real concentration of fecal bacteria in the samples. Plus, this official way to count them does not give information on pathogenicity, okay, potential. So you don't know if they are dangerous or not. You just know you have that number of E. coli and that number of enterococci. You can virtually apply this on all known species or studied species, unless, I mean, you know the nutritional requirements, so what you have to put inside the medium, okay? This kind of approach, so agar medium in a plate, can also be used in a classical or automated version to study antimicrobial resistance, antibiotic resistance. This technique is called antibiotic susceptibility test, AST. And basically what we do is to take a colony, for example, from the previous plate, we let grow a layer of this of copies of this bacterium on an agar plate. So you have the agar medium covered by a layer of microbial growth, and then we put these paper discs. They are very small and they are contaminated with antibiotics, with known amounts, concentration of antibiotics, okay? So this plate is an example of what happened after an incubation time. You will see if a bacterium is resistant or not, or how much is resistant to an antibiotic. Imagine that each of these discs, paper disc with antibiotic, it's a different antibiotic, okay? You have these halos around in case the microbes is sensitive to the antibiotic, so it's not able to grow there, or you don't have the halo if it's resistant. And based on standardized tables on the identification of resistance or sensitivity to an antibiotic based on the diameter of this circle, okay, of this halo, you can say if a bacterium is antibiotic resistant or not, and to what, so to which antibiotic, okay. This is an easy, relatively easy method. You have also automated versions, so they are even faster and even more precise in understanding the resistance or potential or not, because it's not that easy to quantify the diameter, not always. Each bacterial species has its own table of diameters to determine if it's resistant or not. But still, it's a very useful screening test and very relatively cheap to understand if we are dealing with a pathogenic bacterium. For people in Ancona, we do it in the laboratory. If you're interested to see it, we can ask Manu to show you. Okay, and then let's go to molecular application. Most of you have heard about PCR, even people not dealing with molecular biology, polymerase chain reaction. Most of what we do today in biology, environmental biology, microbiology, health, clinical biology, etc., is possible because there is PCR, okay? PCR means that I will try to make it very, very short. DNA is made by a double strand, okay? PCR is a process in which for multiple times, like 35, 40 times, you have this cycle. You have denaturation, annealing, and extension. So you separate the two DNA strands, you let some very short DNA sequence, known sequence called primer, attach to the complementary part in each strand, and you have the extension in which you recreate the complementary part of each strand, okay? So basically, you start from one copy of DNA, and you have, at the end of the cycle, 2 copies. And the number of copies of the target region, which is the one between one primary and the other, will be exponential, okay? Because at each cycle, you will have an exponential number of copies. And this allows you to see if you have amplified the gene, okay? This is an agarose gel, and this is like the classical way we see if we have that target, the target that we need. So if the primer found the complementary part, and if we have the extension of the selected region, okay? This is the classical PCR. We have the so-called quantitative PCR, because we don't need to do the agarose gel. We basically make the reaction using molecules that give fluorescence at each time they create a copy, okay? At each cycle, the more copy you have of the target, the more fluorescence you will have. And the fluorescence is measured by the machine, okay? So these curves are the exponential curves of amplification of a target. And this means that differently from the gel, the agarose gel, you can quantify the initial copies of a gene in your sample. And this is very, very powerful. You can apply this, if you know your target, you can apply this whatever you want, not only microorganisms, everything, RNA, DNA, fish, humans, microbes, whatever. And this is, it's also used a lot in application for microbial pollutants. We have worked with this targeting specific regions of ribosomal genes, both of Enterococci and E. coli. So in a matter of hours, you can get the results of the microbial pollution in a sample. Okay, you know how many copies of E. coli 16S gene you had in your samples. You can also test multiple genes at the same time. So after maybe 1 hour of PCR, you will know if we are talking about, for example, microbial source tracking, you can search for 4 different sources, or if you have fecal contamination by cows, by humans, by dogs, or by birds, and you can have the answer at the same time. After 1 hour, you have all the response to this. It's not paradise, because we would use it all over the world if it was. It's rather expensive, at least the machine. So the first expense is very high. It needs expertise, and it does not discriminate live and dead cells. So what you measure could be also dead fecal indicators, okay? While the plate count method only takes live cells, okay? But I mean, it's very, very powerful, and you will see it can be successfully associated with the traditional methods. Microbial source tracking is using real-time PCR, quantitative PCR. So you can basically look, these are the codes for many genes, but there are like tens of them around the world being developed every day. For viral and bacterial targets to understand the source of contamination. And lastly, the high throughput sequencing. You analyze, maybe you had also a short practical session with Marco to analyze sequence data. Martedì? Sì? No? Practical, yes. Did you? No? Did you see with work? Did you work with R? No. Okay. So basically, sequencing is taking, and I'm talking about 2 approaches: metabarcoding and metagenomics. Metabarcoding means that imagine the filter in which 1 liter of seawater concentrated bacteria. We are extracting DNA from all the bacteria in that sample, and we will analyze all the 16S genes which are present in that samples, which are millions, okay? You have this high throughput sequencing sequence with different approaches, actually, 16S. And then all the work is thereafter to understand what a sequence means in terms of taxonomic identification, okay? It's semi-quantitative. This is the meta-barcoding approach, and it's becoming, I mean, it was considered an expensive approach, is not expensive anymore, so you can get 16S meta-barcoding high throughput sequencing sample per 30 to 50 euros, okay? So it's becoming, it's much cheaper than 10 years ago. So it's untargeted. You can virtually identify all microbes there. It's very useful because all the data that we created, for example, in the last 10 years, can be exploited in the next years to look for new targets that are becoming interesting thanks to research updates, no? And costs are constantly reducing. The disadvantage is that it's not time efficient unless you have your own sequencer in your laboratory, which is not that common unless you are in a university with a service or similar. You still need some days before you get the raw sequence that to be analyzed. It has computational needs, so the more data you will have, and you can get like 200,000 DNA reads from 1 nanogram of DNA, okay, from a single sample of sediment. You need computational power to analyze them. And this is especially true not, for metagenomics, which target not only 16S, but all the genes which are present in a seawater sample, okay, which are millions or billions, okay. So you have much more raw data to be analyzed computationally. Plus, you need, in both cases, bioinformatic expertise and especially for the meta-barcoding approach, you're not, we are not still in the able to reach the species taxonomic level. So most of the time, for the most common meta-barcoding of 16S, the approach that we use, you have fragment of the 16S which is not enough to discriminate between species. So you most of the time you have a list of microbes until the genus level, okay, which sometimes for microbes it's very, I mean, can say a lot of things, but for several scientific questions is not enough, okay. We have tried to summarize which is, I mean, the question was, there is one of these approaches which is better than the other or not? And we compare the same data on microbial pollution, fecal pollution, using culture-based enumeration, using PCR, and using meta-barcoding. And the answer, very briefly, is that you have to combine at least 2 of the approaches in order to have enough information. So enumeration on plate counts is not enough if you don't, for example, you don't see with microbial source tracking, which is the source of contamination. qPCR alone maybe is not giving you information about live versus dead cells, so maybe you are overestimating. And the same is also for 16S high throughput sequencing. So pros and cons which should be considered. And from our side, the response was that to have a very reliable understanding of the contamination, we should use at least 2 of these approaches. Okay, last part. It's too soon. Okay, the last part is very, I mean, I wanted to, you are most likely researcher of tomorrow, okay? So you have to try to familiarize with the fact that you have to write projects, that you have to search online for opportunities to work with scientific teams you are interested in. And plus, since microbiologists here are like minority, I wanted also to show that I mean, think about multidisciplinarity and think about also this morning the prof was mentioning this. You need to understand how to complement your expertise with others. Okay, so microbial pollution can be complementary to oceanography, to a lot of things, and I've learned this during the year. Especially in the place where I'm working now. So we are working a lot on fish microbiome because Irving studies fish, okay? And so this was a great opportunity to understand the functioning of the sea through fish microbiome. So most of the time, what seems very far from you can be a strength in your future career. So this is my good thinking for you, okay? Of course, you've heard about Horizon Europe, for sure. Maybe some of you are already involved in project, or your PhD is within an Horizon project. They are very big projects. You can find, I mean, it's very difficult to dive into the information of the program because you have calls in many moments of the year. For example, I just mentioned here this call, Zero Pollution, in which the topic is decontaminate and bioremediate aquatic pollution. The description of the call is within the link reference to which the call is referenced. And most of the time, for example, microbial pollution can be in this type of calls could be involved. Okay, this is just an example. I'm not going to apply for this call because it's. I mean, it's not the case. But if you want to know more about Horizon Europe, you can, or European initiatives in general, I suggest you to go to the APRE website, which is a national website to inform people in research about the European fundings, European initiatives for a research program. Okay, so you have webinars opportunities, you have frequently asked questions on calls, you have people to ask for specification on particular calls of interest. Okay, and you have a lot of calls. Okay, during the year you have a lot of them. At Irving we are lucky because we have a grant office indicating most of the times new project funding initiatives. But if you don't have, you can start from this website. European Marine Board. I mean, this is, for me, it's very useful to dive into this website. If you don't know it, especially because you are a young researcher, please go there because you can also become ambassador. I mean, you have a lot of opportunities to travel and to create networks with other young researchers, and not only in Europe. The Marine Board makes several, let's say, white papers, so position papers, like this one. They are very easy to read and they are very comprehensive of the status of research, of the priorities in this moment. And in this document, you will find also a part dedicated to ocean and biodiversity, in which microorganism and microbial pollution is mentioned, indicating that this topic is a priority. Not only antimicrobial resistance, but microbial pollution in general in the sea is important also for biodiversity purposes. Okay, you will find many position paper. It's very even for your future, for applications, work position, etc., etc. It's very useful to have a comprehensive idea of what European research is prioritizing. The project that I mentioned about antimicrobial resistance in aquaculture was funded by JPI Aquatic Pollutants. JPI stands for Joint Programming Initiatives. It's European. And Aquatic Pollutants was created by the merging of three JPI's initiatives. JPI AMR on antimicrobial resistance, JPI Oceans, and Water JPI. They merged together to create these aquatic pollutants, which funded several projects on different aspects of aquatic pollution, marine and freshwater. And so it was a very wide container for proposing research on this theme. There was chemical, ecotoxicological, plastic. There was a very wide spectrum of topics funded by this, including antibiotic resistance in aquaculture. Okay, this is now stopped because I hope that they will fund new project in the next future. JPI and AMR, JPI Oceans, and JPI Water are working. They fund the project. I'm involved in a JPI Ocean project in this moment. And they also make a lot of initiatives, also in this case, webinars and other things that could be very useful for you also at this stage of your career. Okay, also UNESCO. I mean, I found this at the wider level, not only European. Ocean conservation, ocean health is a topic at the global level, even if we are in an historical moment in which everything is it's not always believed it's true. We are in emergency. Okay, you are a researcher. You know that we are in an emergency on the planet. Okay, and so there are also global initiatives on this. There is the Ocean Decade, which is something that maybe you have heard about. And in this page, I found a very interesting Excel file you can download here, in which there is a list of projects endorsed, whose research is endorsing the Ocean Decade action here. You will find the people involved, the topic, the link for the project, and if it's something that it's interesting for you, maybe also get in contact with people working on this. And that's all. Thank you very much. If you have questions. I cannot answer to this question, but I can say that at the university, at the Politecnica, I had the opportunity to listen to a very interesting seminar by professor. I will tell you. I have the name. She's an engineer, I think, and they are working a lot on combined sewer system. And she also provided in this seminar several current knowledge, and several solutions at the engineeristic level on this. So this is, too much different from my knowledge. I'm quite sure that there are other countries, especially I'm quite sure in the northern part of Europe, dealing with this. But if we are able to improve our infrastructure here, it would be, a big good news for us. Okay. So the problem is that the quantity of water that we are receiving by weather events in the last years, it's a lot higher than we used to deal with in the last year. So we are experiencing very intense events, and we were not ready for this. This is my opinion. So the infrastructure is not enough to deal with the impact, with the increased quantity of water which is reaching the system. That's one point. Smart things, I'm sure, but I'm not aware of. Scrivela tu. Allora, magari chiedo aiuto a...
Perché faccio